name is Mariah. I'm Preserves and Volunteer Manager, if you haven't met me before, on the Little River Wetlands Project. And of course, our mission, if you don't know it, is to protect and restore wetlands in the Little River Valley, along with educating the public on how to be good stewards of the land. So we are really honored to have Dr. Kingsbury here today. He came to then IPFW in 1992 after obtaining his PhD from the University of California, Riverside. Recently, he served as the Associate Dean of the College of Arts and Sciences, which is now the College of Science and College of Liberal Arts. Prior to that, he served 2 terms as Chair of Biology. Dr. Kingsbury also directs the Environmental Resources Center, one of the university's centers of excellence. The mission of the ERC is to promote the understanding and conservation of our natural resources. Bruce and his students conduct research on the ecology, behavior, and conservation of wildlife, especially imperiled reptiles. His work has been supported by over $3 million in external grants and has resulted in numerous publications and presentations. So we're super lucky to have him here today, and what an interesting talk, Valentine's themed. So thank you, Bruce.
All right, so I first gave this a part of this talk to the Herpetology Club last year when they asked me to do something Valentine-y. And so then I've expanded that for you. But I'm going to talk about hearts, but as I got into hearts, I can see that I need to tell you a little bit about the circulatory system, and I'm going to try to resist talking to you about respiration because they're all tied together, and the plumbing is all kind of connected. But that's kind of my idea. And then here's a kind of the classic thing. We've all seen this in school or something like this. This is a mammal heart with atria are taking blood from the body, delivering it to the right ventricle. That's pumping it out to the pulmonary arteries where it gets oxygenated, and then it comes back to the pulmonary veins, to the left atrium, and the left ventricle pumps it out to the body. And then there's these different branches that go out towards the head and the arms and then down to the rest of the body. So that's kind of the standard, but that's not at all what's always there. So it's kind of the fun part of it is like, what else is there? And so to get into that, I thought I'd start with insects because they provide a good comparison in the sense of they have multiple hearts. The hearts are not in the same place as ours, and they have what's called an open circulatory system. And so you have hearts back here in the abdomen, and they just have slots that open into the body, and that each of those has a valve that promotes one-way flow into the heart. So and then they pump the blood forward, and then it just diffuses out into the body again, and the hemolymph in the body is not, there's no capillaries, there's nothing at all like that. So that is very different. The other thing that's interesting for organisms like this is the way that they obtain oxygen and get rid of carbon dioxide is, I don't know if you've seen this before, but along the abdomen they'll have little openings called spiracles, and those open to these tracheae, these tubes, and those go into the body, and that's it. There's no lung, there's no gills, and so although you see large insects in horror movies, it's actually physically impossible to get a bug that's bigger than about that. And the reason why is that they are constrained by gas exchange. They just can't transfer oxygen in and carbon dioxide out fast enough. But they have hearts. Yeah, more than one. And while we were here, we were going to check out the respiratory system. And then here's a spider for comparison, but the premise is basically the same. You have, here your heart has these multiple slits in it, and it pumps the blood into arteries. Again, there's no veins. They do have a lung that's kind of like a modified gill, where there's gas exchange. But if we look far enough back systematically, there isn't a structure like we see with mammals. And then with something like an earthworm, we have a modification of where the hearts are, but we have another advance, which is a closed circulatory system. So they have 5 pairs of hearts up here, and then they do have arteries and veins and capillaries and that kind of thing. So if we look at something like that, we at least see a closed circulatory system. All right, there, we've done invertebrates, we're done. So now let's look at tetrapods. I do see some chats. We better take a quick look at that. And then I think I can hide this panel. And that way we can see around. Oh, go away. Okay, so now if we look at tetrapods, that's 4-legged things, and so this is also, just for background, kind of like the classic plumbing system that might then be modified. So the blood comes from the lungs into the left atrium, the blood comes from the body into the right atrium. Here it's pumped to the heart through the right ventricle, pumped from the left ventricle out into the body. Carotid arteries go to the head, and then the dorsal aorta goes down the body and services different parts. What's also interesting about this, as an aside, is that the liver and the digestive tract, the kidney, and then the body and the legs all have their own dedicated plumbing. So it's not like the blood has to go to the legs first and then stop over by the kidney and then do the liver on the way out. And that means that it's much more efficient in terms of delivery of oxygenated blood everywhere in the body. But that isn't the way that it always is. So that's the reason I'm showing it to you, is because it's not the way it always is.
What would be an example of a tetrapod?
Us, frogs, snakes. It's basically a reference to all of the creatures that evolved from fish and moved onto land, and they have legs.
But not necessarily a mammal?
It would also include mammals in it. And the way we use this terminology, it's referring to what we call unique derived traits. So that is when we have something like a primitive amphibian moving onto land, we might then have a mammal be bipedal, snake not have any legs, but they're still tetrapods because of their origin.
Oh.
All right, so there's the basic plumbing. And we lost that again. So let's go back over here. Technology is great when it works. So this is a fish, and fish are not the same. And fish are not tetrapods. Tetrapods are derived from fish. And there are lots of different kinds of fish. This is kind of a generic schematic of them. But the heart is quite different. There's an initial chamber called the sinus venosus, which basically sort of organizes the blood and delivers it to the atrium. And then that delivers it to a ventricle, and then that goes to the gills. So you have like 2.5 chambers in this, not 3, not 4, but and keep in mind, because this will be kind of important later, they have gills, they don't have lungs, and so the plumbing is going to be different. And then once the blood leaves the gills, it can go by the carotid arteries to the head, or via dorsal aorta out to the body, and then things are collected and sent back. And I want to say this now, I have it on a slide later, but there will be a quiz. And if you don't get 70%, you have to do this again, over and over again until... All right, so here is the gill structure up here. And there's some detail on this that basically you don't need to worry too much about the detail, but I want you to sort of see the high elevation vision of this. And if you think about sharks with where you can see the gill slits on the side, basically you have a similar sort of structure in a bony fish, except they have that operculum that covers it up. But the structure inside of there is you have a bunch of gill arches, and between them slits, and a blood flow all through those gills, so that you drop your CO2 and you pick up your oxygen, and then some of the blood goes up towards the head, and some of the blood turns and goes down to the rest of the body. So that plumbing is kind of different too. But again, the heart structure is pretty simple. And in reality, this is sort of shaped like an S with the ventricle in the bottom and the atrium stacked on the top. But just to look at the flow, this is a convenient way to look at it. All right, so that is a little bit on fish. And for the rest of the presentation, I want to just sort of tease out some different kinds of hearts and parts of the circulatory system and compare things like frogs to mammals and stuff like that. And I joked that the presentation was 3 hours long, but it's not. This isn't a big, long presentation. So frog hearts have 3 chambers, 2 atria, and 1 ventricle. And but they have the same number of jobs basically that like a mammalian heart would have, and so there has to be some trickery involved. So the blood comes into the left atrium from the body, and the right atrium, which is on the either side over here, from the lungs. But then it gets to the ventricle and they mix. They don't fully mix because there's some structure in the ventricle which keeps the flow, you could say laminar, but the pathways of the blood are right next to one another. So there's not a complete mixing, but they're actually right next to one another in there. And then the flow then goes out to a big set of blood vessels going out to the head and the rest of the body. Now, frogs and turtles and crocodiles do different things during life. And for something like a frog, a lot of times they are doing a lot of gas exchange from their skin. They're underwater, they're not breathing. So how do they do that? So when they are underwater and they're not breathing, the volume of blood that's that's going to and from the lungs is reduced, and so all of the plumbing that has to do with delivering blood to and from the lungs is also minimized. It's still there, but it's just not full. And so the blood that comes in just goes right back out to the body instead of to the lungs. And part of that blood is delivered more preferentially to the skin, and that allows for gas exchange. Now, the coolest thing for me when I was getting the talk ready for the club last year was, what I realized was that something weird has to happen during metamorphosis for a frog, and actually probably if you looked at the metamorphosis of any kind of an organism, similar things would be happening. A tadpole has gills, and an adult does not. Adult has lungs, and whenever I had historically talked about metamorphosis, I would highlight the fact that you're resorbing the tail, you're budding legs that weren't there before, and there's all these fascinating changes. But in fact, stuff's happening inside as well, and it has to do with circulation, among other things. And so here is what's going on in a tadpole, and the symbology here is, if it's purple, the blood is a mixture of oxygenated and deoxygenated blood. And you'll, when we have it, you'll see that there's oxygenated blood, and that would be stuff that's leaving the lungs and going out to the body, actually leaving the heart oxygenated, or coming back from the body deoxygenated. But here, it's all mixed. So tadpoles are not going to be very athletic. But you have blood coming back from the head, blood coming back from the body in various sorts of ways to the right atrium. Auricle is just another name for atrium. Collected at the ventricle and delivered actually through a thing called a conus arteriosus. That's what this other thing is, but it has a valve in it that distributes the blood. It's a muscular spiral that sends the blood out here. But some of this blood goes to the head before it even crosses the gills. And where the blood from the gills goes is in different directions. And the end result here is that none of the blood is perfectly oxygenated, and similarly, none of it is perfectly deoxygenated. So when I was talking to you about the circulatory system of mammals, or the heart of mammals, we are very good at splitting those apart so that when we're delivering blood, say, to the brain, that's as oxygenated as we can make it. That isn't true with a tadpole. And that's why we call it mixed. And then as they move into using lungs, they don't have gills anymore, So you need a complete rearrangement of those blood vessels, because now you need to send blood to an organ that wasn't even there. And you need to not deliver it someplace where there are no gills anymore. So while the frog is struggling with growing legs and resorbing a tail and switching from algae to insects, and mouth parts are changing, all that, so is the heart, so are the blood vessels. And you start to have separation of oxygenated and deoxygenated blood. And so here's the vessels that are going up to the head. They have oxygenated blood. The blood that's going to the body is being, just like with the frog I showed you before, is being split again. And even though the flows are side by side here, you do have a tendency to deliver blood that is deoxygenated to the lungs and the skin, and oxygenated blood out to the body. But still, a very simplistic structure compared to what we have. And I mentioned tetrapods. Another term I can throw in there is the notion of an amniote. Amniote refers to the amniotic egg, which is the kind of egg that we would have, and we have another layer in the egg, which allows to have the amniotic cavity. And so when you hear about amniotic fluid, like if we're sampling that for testing or something like that, that's in a space in the embryo that wasn't there in frogs and isn't there in fish, but it is there in reptiles and birds and mammals. And so when I talk about amniotes, I'm talking about reptiles, birds, mammals, that kind of stuff. And to get towards the human heart as maybe our kind of our end goal, we have to get rid of all kinds of the plumbing that used to go to the gills, and what you'll see is evolution. When you watch evolution, what you see is the reuse of stuff that's already there, or the removal of things that don't get used anymore. So it turns out actually that the blood vessels that go to the head are modified from remnants of those that was what was going through the gills, and then same thing down here, and you end up with a simplified system, which will even be more simplified as we go. But the thing that I wanted to highlight here was this conus arteriosus that I mentioned before. It's just a spiraling piece of tissue inside this little bit of a chamber, and that is what actually keeps the blood more effectively from mixing. And so you are delivering blood that's less oxygenated to the lungs, and delivering blood to the body that is more oxygenated. So it is actually, we're getting gradually more efficient. And now what I want to show you is that if you look at reptiles and birds and mammals, it turns out that even those are different. So we're starting out with a sort of a generalized heart and actually modifying it depending on the taxonomic group. And the thing that I wanted to show you here is, those of you who know some anatomy know about our descending aorta is one blood vessel. Well, in other organisms, that's not true. There's actually left and right sides. And so here, we actually have these two vessels, one of which is more oxygenated, and that goes to the head and then down to the body. But actually, there's another one that comes out of the heart and goes down to the body. And that's has mixed blood in it because it's being delivered between the arteries that are going to the lungs and the arteries that are going out to the body. So that's our sort of what I'm calling a generalized reptile, sort of a reptile heart cartoon. And so with crocodiles are interesting because they actually have a 4-chambered heart. So you'll notice that a lot of these other organisms don't have 4 chambers like we do, but crocodiles do. And what that means is that the septum that is between what could be a right and left ventricle actually gets more and more developed until you have 2 chambers. And in crocodiles, it's fully developed. It's not the same as our heart, but the result is 4 chambers. But crocodiles spend a lot of time underwater. They hold their breath for long periods of time. So they have kind of the same sorts of challenges that a frog underwater would have, or a tadpole. And so their solution is this foramen of Panizza, which is a little window right here between these 2 arteries. And if they are underwater, they reduce the flow of blood that goes to the lungs, but they can redirect it between these vessels so that it still sends blood everywhere. So what you see over and over again is when the animal is underwater, they have to change the blood flow inside of the heart. Turtles. Now turtles don't have 4 chambers, but the septum between the sides of the ventricle is relatively well developed, and that allows them to tweak some stuff. So here's basic flow for a turtle. So the sort of the lub-dub that you hear in a heartbeat, the first thing you hear, you're actually listening to valves opening and closing. But here I'm inferring what happens in the first part of the pulse where you're draining the atria, and the second part where you're draining the ventricles. And basically, in all hearts, what the atria do is collect blood into kind of a package and then deliver it down to the ventricle. Here you go. And then the ventricle then is the really muscular part that shoots it into the rest of the bloodstream. So here we have blood from the right atrium going into the ventricle. Here we have the left atrium, more blood going into the ventricle. But this blood is from the pulmonary veins, so it's oxygenated. And this blood is from the body, so it's deoxygenated. So now you have those 2 kinds of blood in the ventricle at the same time. And then you have the contraction of the heart and of the ventricle, and then you have this muscly divider, which is not, there's a big hole here, but it's enough to redirect the blood to the appropriate location and keep the 2 types relatively separate. So here the atria contract, loads the ventricle. Here the ventricle contracts and it delivers the blood to the lungs and the skin and the head and the rest of the body. So, of course, turtles hold their breath too. So what they do under those circumstances is again reduce the activity on the side of the vascular system from going to and from the lungs, and emphasizing instead the blood that's coming from the body, and then the blood just flows around like that, and that way, even though the lungs, there's nothing going on in the lungs, so you send the blood this way, and it still completes the circuit and sends the blood out to the body. So you keep pumping it around. What about birds and dinosaurs? Reasonably, we can say that birds are essentially dinosaurs. What they are is what's left of a lineage of reptiles, which also included the dinosaurs. So turtles are their own thing. Dinosaurs and birds are another lineage. Lizards and snakes are another lineage. And actually, mammals are their own lineage of another group of reptiles. So those are all different groups. So what's nice about that, and the reason I mention it, is you can look at what birds do, and that's your best look at what dinosaurs would have done. And they have a four-chambered heart. And actually, by the way, crocodilians are somewhat related to these guys too, just way back when. But here's a four-chambered heart. That left, remember that left systemic artery that I had plugged in the middle here, that's gone. And it's a four-chambered heart. Now there's no mixing. But interestingly, in birds, they emphasize this as their aorta. So the right systemic artery. What you'll notice is, for us, it's the left. So there's a solution, but there's a need, but the solution is different. And so now, the blood that goes out to the lungs is the deoxygenated blood. It gets reoxygenated, it comes back to the heart, and then the blood that's going out to the body and the head is fully oxygenated. So that's nicely divided. And on our way to mammals, the group of reptiles that mammals come from are the synapsids. So here's the model there. They have both of these aortas, basically the systemic arteries, and it's all connected up here. Notice how everybody comes out one time and then goes everywhere, to the head, to the arms, down to the rest of the body. So that's you can see that that's different from the birds. So we're going to take this and we're going to modify it a little more to get to this. And then here's mammals. Now you can find all the parts that we just looked at, say in this one, but where they come from is different. So now that right systemic artery that we saw in birds isn't there anymore, but the left one is. So birds went this way, mammals went this way. But they had a conference in Geneva and they decided they didn't need both, and they could choose which one they wanted. Oh, so they did. So maybe I should have brought us back to the mammalian heart at the end. But that's what I have for you today regarding the hearts, and I hope you had fun. But I hope you could see that the solutions to circulation are not all the same depending on what animal you're looking at, and that as we have this divergence of different groups, different major taxonomic groups, we have these patterns so that mammals are one way, birds are one way, crocodilians are kind of also doing their own thing, etc. So here I'll open it up for questions and discussions. I did throw in this image because for those of you who enjoyed the conservation conversation that we had before, we're gonna start doing that again. But because we do a lot of things like with Little River and other groups around Earth Day, we thought we would move it to the fall. So that's coming again. I didn't actually send around this sign-up sheet. I know some of you are already on that, but if you are so inclined to learn more about the ERC, we have we do have a newsletter. It comes out every month, and so if you're not signed up for that and you're interested, please do sign up there. Also have our website here and the address. And I didn't do my usual plug for the ERC. I think a lot of you already know about us. But certainly I'm happy to collaborate with Little River on a variety of things. And we, as Mariah said, we do understanding conservation of our natural resources, and events like this are some of what we do. It's totally different from that is the kind of research we do. We study turtles and snakes and birds and frogs, and other faculty study bats and all kinds of things. But that's a kind of another part of what we do. So thank you.